Okay, guys, so today we're going to finish up on your frog dissection lab, and then you should be finished and can submit your answers to the questions. So here we go.
Digestive system.
In this lesson, you will do the following. Trace the path food takes through the alimentary canal. Identify digestive organs and learn of their function. Remove the liver and alimentary canal. Prepare a microscope slide of villi and examine it.
Digestion in the frog starts at the mouth, so let's open it. Frogs will eat almost anything, but usually prefer insects. In fact, their tongue is well adapted at catching small prey like this. Press on it. As you can see, the tongue flips forward and is hinged at the front. This allows it to flip prey back into the mouth. Find and click on the vomerine and maxillary teeth. Although the frog has maxillary teeth and tiny vomerine teeth, it uses them more for grasping than biting or chewing. The objective for the frog is to get as much of the prey into its mouth as it can. Once prey is fully inside, the strong rolled muscles belt the prey down the gullet. Press on the gullet to continue. This is the beginning of the alimentary canal, which is a continuous canal made from the esophagus, stomach, intestines, and cloaca. The gullet's job is to close off the alimentary canal when the frog pumps air into its lungs, and also perhaps to keep food from escaping back up its throat. Just past the gullet is the esophagus, which uses peristaltic contractions to squeeze food down to the stomach. Lift up the liver and press on the stomach beneath it. Inside the stomach, strong acids and more muscle contractions break the food down into a soupy consistency. Click on the pylorus. The soupy mixture is temporarily stored here until the pyloric sphincter at the end of the pylorus opens into the duodenum. Click on the duodenum. In the duodenum, a duct from the pancreas and gallbladder deposits special enzymes into the soupy mixture. To find out what these enzymes do, press on the gallbladder underneath the liver. The liver makes a fat-dissolving enzyme called bile, which is stored here. Ducts from both the liver and gallbladder form the common bile duct, which passes through the ribbon-like pancreas. Now click on the pancreas, wedge it between the stomach and duodenum. The pancreas produces more digestive enzymes, including an acid-neutralizing enzyme. Without it, the digestive mixture from the stomach would burn the walls of the intestines and eventually destroy them. Click on the small intestine. Once the acid is neutralized and fat is broken down, the resulting mixture is passed through the lengthy small intestine, where nutrients are absorbed. Tiny projections of epithelial tissue called villi do the absorbing, which line the walls of the small intestine. Capillaries collect the nutrients and send them up through veins in the thin membrane of the mesentery. These nutrients then move up the hepatic portal vein to the liver, where they are converted into glycogen. Press on the spleen at the bottom of the mesentery. The spleen breaks down old red blood cells and is actually part of the lymphatic system. However, the pigment from these cells is also sent up through the hepatic portal vein to the liver, where it is used in the formation of bile. Find and press on the large intestine. The large intestine, which is also called the colon, absorbs excess water and compacts the mixture into a moist solid. With the aid of helpful bacteria, a final bit of nutrients are also absorbed here. The leftover feces is then forced into the cloaca for disposal out of the body. Select the scissors on the toolbar. Cut away the liver, esophagus, and large intestine. Place the liver to the right. Now place the cut portion of the alimentary canal to the right, and pin it. Select the scalpel on the toolbar. Create a slide sample of the small intestine by slicing it twice, and then press the forceps button. Place the cross-section on the microscope slide, and press the microscope button. Focus the view by pressing the white dot and turning the dial clockwise. As the image comes into focus, you will see that villi create a greater surface area to absorb nutrients. This should be made clear by comparing the length around the inner surface and outer surface of the small intestine. Press Done to continue. Select the scissors and cut open the stomach. Now open it. You have completed this lesson.
In this lesson you will do the following. Identify the kidneys and reproductive organs of a male frog. Identify the female's reproductive organs and observe their processes. Learn how direct external fertilization occurs. Examine and observe the development of a fertilized egg.
The urogenital system is the combination of the renal and reproductive organs working together. This relationship is most apparent in the male bullfrog. Click on the kidneys. Each kidney is comprised of long thin tubes, called tubules, that are covered in capillaries. These vessels filter blood of nitrogenous wastes, which in turn form urine inside the tubules. On male bullfrogs, the testes are attached on the ventral side of each kidney. Click on one. The testes are dependent upon the kidneys to transport sperm through their tubules. A mixture of sperm and urine drain into the archenephric ducts at the bottom of both kidneys. Find and click on one of these ducts. Each archenephric duct deposits urine into the cloaca and separates sperm into a chamber called the seminal vesicle at its lower end. Press on the seminal vesicle. The urine that enters the cloaca is stored within the lower abdominal cavity in a sac called the bladder. Press on it. Urine is kept here until the frog decides to discharge it back through its cloaca and out of itself. On the side note, you may see orange-colored projections emerging around the testes in males and ovaries in females. These are the fat bodies. Click on them. The fat bodies are used for storing energy and can vary in size from small projections like these to massive ones that fill up the cavity. Now let's take a look at the female frog's reproductive organs. The first organs you might notice are the large black ovaries that attach on the ventral side, the kidneys. Click on one of them. Unlike the male's testes, the female's ovaries release gametes directly into the abdominal cavity through its own membrane walls. Press on the scissors button and cut away the ovaries. Place the ovaries to the right. With the aid of current-producing cilia lining the abdominal cavity, the released eggs move upwards towards the tips of the oviducts. Press on an oviduct. Each oviduct tip also has cilia lining its opening in order to draw in the eggs. These tips are each called a tubal ostium. Press on one. When the eggs pass through the tubal ostium and lengthy oviduct, they are coated with a protective jelly and stored in the uterus. Press on the uterus to continue. During mating, a male piggybacks the female in the water. This is called amplexus. Here the female releases her eggs into the water from each uterus. Simultaneously, the male releases the sperm stored in his seminal vesicles onto the eggs. This method is called direct external fertilization, and is usually repeated with more than one male. To see what happens after fertilization occurs, press on the microscope button and focus the view. This is a 6-day time-lapse simulation of a tadpole forming from a newly fertilized egg. When you are through with your observations, press the Done button. You have now completed this lesson.
Okay guys, there's going to be one last video for the nervous system and then you'll be done with this assignment.